Postovulatory follicles are structures formed after oocyte release. They do not have endocrine function, present a wide irregular lumen, and are rapidly reabsorbed in a process involving the apoptosis of follicular cells. A degenerative process called follicular atresia reabsorbs vitellogenic oocytes not spawned. This process can also occur, but less frequently, in oocytes in other development stages. Some fish, like the California sheephead, are hermaphrodites, having both testes and ovaries either at different phases in their life cycle or, as in hamlets, have them simultaneously. Over 97% of all known fish are oviparous, that is, the eggs develop outside the mother's body. Examples of oviparous fish include salmon, goldfish, cichlids, tuna, and eels. In the majority of these species, fertilization takes place outside the mother's body, with the male and female fish shedding their gametes into the surrounding water. However, a few oviparous fish practice internal fertilization, with the male using some sort of intromittent organ to deliver sperm into the genital opening of the female, most notably the oviparous sharks, such as the horn shark, and oviparous rays, such as skates. In these cases, the male is equipped with a pair of modified pelvic fins known as claspers. Marine fish can produce high numbers of eggs, which are often released into the open water column. The eggs have an average diameter of 1 mm, 0.039 in. The newly hatched young of oviparous fish are called larvae. They are usually poorly formed, carry a large yolk sac for nourishment, and are very different in appearance from juvenile and adult specimens. The larval period in oviparous fish is relatively short, usually only several weeks, and larvae rapidly grow and change appearance and structure, a process termed metamorphosis, to become juveniles. During this transition, larvae must switch from their yolk sac to feeding on zooplankton prey, a process which depends on typically inadequate zooplankton density, starving many larvae. In ovoviviparous fish, the eggs develop inside the mother's body after internal fertilization but receive little or no nourishment directly from the mother, depending instead on the yolk. Each embryo develops in its own egg. Familiar examples of ovoviviparous fish include guppies, angel sharks, and coelacanths. Some species of fish are viviparous. In such species, the mother retains the eggs and nourishes the embryos. Typically, viviparous fish have a structure analogous to the placenta seen in mammals, connecting the mother's blood supply with that of the embryo. Examples of viviparous fish include the surf perches, split fins, and lemon shark. Some viviparous fish exhibit oophagy, in which the developing embryos eat other eggs produced by the mother. This has been observed primarily among sharks, such as the shortfin mako and porbeagle, but is known for a few bony fish as well, such as the half-beaked Nomaramphus ebrardtii. Intrauterine cannibalism is an even more unusual mode of vivipary, in which the largest embryos eat weaker and smaller siblings. This behavior is also most commonly found among sharks, such as the gray nurse shark, but has also been reported for Nomaramphus ebrardtii. Aquarists commonly refer to ovoviviparous and viviparous fish as live bearers. Acoustic communication in fish involves the transmission of acoustic signals from one individual of a species to another. The production of sounds as a means of communication among fish is most often used in the context of feeding, aggression, or courtship behavior. The sounds emitted can vary depending on the species and stimulus involved. Fish can produce either stridulatory sounds by moving components of the skeletal system, or can produce non-stridulatory sounds by manipulating specialized organs such as the swim bladder. There are some species of fish that can produce sounds by rubbing or grinding their bones together. These noises produced by bone-on-bone interactions are known as stridulatory sounds. An example of this is seen in Hemulon flavolineatum, a species commonly referred to as the French grunt fish, as it produces a grunting noise by grinding its teeth together. This behavior is most pronounced when the H. flavolineatum is in distress situations. The grunts produced by this species of fishes generate a frequency of approximately 700 Hz, and last approximately 47 milliseconds. The H. flavolineatum does not emit sounds with frequencies greater than 1000 Hz, and does not detect sounds that have frequencies greater than 1050 Hz. In a study conducted by Oliveira et Alabama, 2014, the long-snout seahorse, Hippocampus reidae, was recorded producing two different categories of sounds, clicks and growls. The sounds emitted by the H. reidae are accomplished by rubbing their coronet bone across the grooved section of their neurocranium. Clicking sounds were found to be primarily produced during courtship and feeding, and the frequencies of clicks were within the range of 50 Hz, 800 Hz. The frequencies were noted to be on the higher end of the range during spawning periods, when the female and male fishes were less than 15 centimeters apart. Growl sounds were produced when the H. reidae encountered stressful situations, such as handling by researchers. The growl sounds consist of a series of sound pulses, and are emitted simultaneously with body vibrations. 50SOME fish species create noise by engaging specialized muscles that contract and cause swim bladder vibrations. Oyster toadfish produce loud grunting sounds by contracting muscles located along the sides of their swim bladder, known as sonic muscles. Female and male toadfishes emit short-duration grunts, often as a fright response. In addition to short-duration grunts, male toadfishes produce boat whistle calls. These calls are longer in duration, lower in frequency, and are primarily used to attract mates. The sounds emitted by the otow have frequency range of 140 Hz to 260 Hz. The frequencies of the calls depend on the rate at which the sonic muscles contract. The red drum, Cyanops ocellatus, produces drumming sounds by vibrating. Its swim bladder. Vibrations are caused by the rapid contraction of sonic muscles that surround the dorsal aspect of the swim bladder. These vibrations result in repeated sounds with frequencies that range from 100 to greater than 200 Hz. The S. ocellatus can produce different calls depending on the stimuli involved. The sounds created in courtship situations are different from those made during distressing events such as predatorial attacks. Unlike the males of the S. ocellatus species, the females of this species don't produce sounds and lack sound-producing, sonic, muscles.